Good morning, good morning, good morning. Good morning, good morning, good morning.
Okay, so let's do just a final recap with this diagram. The different things we have now studied are glycolysis, the Krebs cycle, oxidative phosphorylation, where we studied the electron transport, and the last thing we saw in the last video were the two types of fermentation: lactic fermentation, which produces lactic acid, and alcoholic fermentation, which produces ethanol. What I would like to point out is that here in the Krebs cycle are produced NADH2 and FADH2, NADH2 here, and same thing in glycolysis. And these NADH2s, they'll be used in the electron transport chain. So indirectly, these NADH2s, they will be able to produce ATP. So the first thing we're going to do is compare the energetic efficiency of fermentation on the one side versus cellular respiration on the other. Let's do that. Number 9, aerobic versus anaerobic respiration. Anaerobic respiration is another word for fermentation. Ana means without air, without oxygen. And we're going to compare the ATP production. As I said earlier on, the NADH and the FADH will be then used to produce ATP. So what we're going to have a look at is the total amount of ATP which is produced. So for respiration, you can read it here. It's between 36 and 38 ATPs. So let's say 36. It's not a round number because the way NADHs and FADHs are going to be used, it's not going to produce a round number of ATP. It's going to vary between, so as you see for glycolysis, between 4 and 6, and therefore on the whole it's going to vary between 36 and 38 ATPs. So respiration at the minimum will produce 36 ATPs, whereas fermentation is only going to produce the ones which are produced in fermentation. That is why we're going to have 2. So in terms of efficiency, when you use up 1 molecule of glucose, with fermentation 2 ATPs are produced, whereas with respiration 36 ATPs are produced at the minimum. 36 divided by 2 is 18. So we can say that respiration, if we from the glucose use perspective, is 18 times more efficient than fermentation. Respiration with the same amount of glucose will produce 18 times more ATP, 80 times more energy. 9.2, the Pasteur effect. The Pasteur effect is a very simple principle. Pasteur was doing experiments and he realized that yeasts were capable of either doing respiration, cellular respiration, or they were capable of doing fermentation. So the Pasteur effect is this capacity that yeast have to switch between aerobic and anaerobic respiration in yeast. We're going to have a look at the experiments that Pasteur carried out to see why he was able to conclude that yeast were able to do both. Now then, we're going to have a look at the experiments that were carried out by Pasteur. There are 2 things that we're going to look at: the alcohol production you can see that there is none at the beginning, and the more we go through the experiments, the more alcohol is produced. We can translate this by the fact that if there is more alcohol produced, that means there is more fermentation. Because fermentation, you know this now, it produces ethanol. So if there's more ethanol, more alcohol, then there has been more fermentation. The other thing is the glucose intake. So how much glucose is used by the yeast. That's what we have on the right, but we'll mainly focus on the left column. Let's now have a look at the different conditions in which these experiments were carried out. So number one, the yellow line that we have at the bottom, this yellow line is a glucose solution. And on top, what we have here is a yeast layer. So in this opened container, there is a glucose solution at the bottom, and on the surface, there is a layer of yeast. In this experiment, no alcohol is produced. We can deduce that there is no fermentation which is occurring. This seems logic because we've got lots of air, therefore lots of dioxygen coming into the medium, so yeasts are doing respiration. Second experiment, glucose solutions plus 2 yeast layers. So at the top there are 2 yeast layers this time, and at the bottom there is still the glucose solution. There's a little bit of alcohol that's been produced. We can analyze this by saying that perhaps the 2 layers, they block a little bit more the O2 molecules from going, for example, to the second layer. Therefore, perhaps the yeasts on the bottom, they will be doing fermentation a bit more, but overall it will be still mainly respiration. Okay, next setup. You can see that Pasteur has changed container this time. In the first ones we had open containers which had direct contact with the air. In this case, the only contact is here through this incurved tube. Inside the flask or balloon we have a glucose solution and 1 layer of yeast. The fact that this curved tube and this closed flask have been used, that means the air is hardly renewed. And we can see that the alcohol production has increased. Therefore, when there is less air coming in, there is more alcohol. So when there is less air, yeasts seem to switch towards fermentation. Let's have a look at the next setups. Number 4, we still have the glucose solution in the bottom, a yeast layer, which is, in terms of amount, is the same as this previous one. But this time you can see that the flask is completely closed. That means the air is not renewed. And we can see that the alcohol production has increased even more. So we're starting to get this idea that when yeasts are less in contact with air, they will do less respiration, less cell respiration, and they will do more fermentation. That's why more alcohol is produced. So let's have a look at the last 2. Number 5. So now the flask is completely filled up with a solution which has glucose. The amount of yeast which have been mixed with the solution is the same as previously, but as it's filled up with the solution, there is less air than in the previous one. In the previous one, we had almost half the flask which had air, and therefore a little bit of dioxygen. In this one, there is no air, So that means even less dioxygen, and the alcohol production is even greater. So again, we have this idea that when there is less and less air, that means less and less dioxygen, and therefore the yeast are producing even more alcohol. They're doing even more glucose. They're doing even more fermentation. And the last one, you may wonder how on earth we can put even less air. Well, Pasteur found a solution to take the air out of the solution. I don't know if you know this, but gases will dissolve into solutions in water, for example. Well, he found a way to take this air out. Therefore, in this last experiment, we still have a glucose solution which fills up the flask, but this glucose solution has no air in it. The amount of yeasts mixed up is the same, and obviously it's still closed, and we can see that the alcohol production is even greater. So that's how Pasteur concluded that the less air, the more alcohol is produced, therefore the more fermentation yeasts do. But as it's the same yeasts, we can see that when they have access to O2, they're quite happy to do respiration, and they don't do fermentation. So there must be a system which actually blocks fermentation from occurring when O2 is present. That means that O2 maybe binds to something and prevents yeast from doing fermentation. It's logic from a natural selection point of view, because we saw previously that respiration is much more efficient in terms of energy production. So if you have a medium with a little bit of glucose, it's much more efficient for the yeasts to do respiration. So we can imagine that throughout natural selection, yeasts which were more capable of inhibiting fermentation in the presence of O2 was selected, and that's how the mechanism was probably selected. The last thing we can see is that the less dioxygen, so there is more and more fermentation going on, more and more alcohol produced, but there's also more glucose that's been taken by the yeast. So for the yeast to maintain the same metabolism, to maintain the same lifestyle, if I may say so, they need to use much, much more glucose when they're doing fermentation. That's why it seems logic that there's this inhibiting system that when O2 is present, respiration takes over and stops fermentation from occurring. Okay, what I'm going to tell you now, you don't need to know for the baccalaureate, but I just wanted to give you some scientific ideas to how this Pasteur effect works. The first thing you need to know is something called allosteric inhibition. You have an inhibitor which will bind to a allosteric site, and when this molecule binds, that will have a consequence to change the active site. So the active site will have a first shape before the molecule binds, and once the molecule has bound to the enzyme, it changes shape. Therefore, the substrate can't bind to the enzyme anymore, and therefore the reaction where the substrate is transformed into products can't occur anymore. So when there is dioxygen in the medium, there is a lot of citrate and ATP produced by the Krebs cycle and the oxidative phosphorylation process, and they're going to inhibit, so they're going to modify the third enzyme, which is, oops, sorry, that's not the enzyme. They're going to modify the third enzyme, which will act here. So you remember that all of these steps, they are speeded up by an enzyme. So enzyme 3 here, this enzyme will have this allosteric inhibition by ATP and citrate. Therefore, less pyruvate will be produced, and the very few pyruvate molecules which will be produced will be mostly used in the Krebs cycle, because they will have the Krebs cycle will have the molecules and reactions will have a bigger affinity for pyruvate than what happens with fermentation. So this is the few elements to help you a little bit understand how this Pasteur effect works.
Fermenting is a reaction. Oxygen levels high, some types of bacteria and yeast. Oxygen levels high, fermentation produces alcohol. Oxygen levels high, some types of bread are made by using yeast.